Today we're diving into the most fascinating system in our body, the immune system. This is the system that stands guard every second of every day, protecting us from the world full of threats. We'll explore not just how it works, but also how sometimes it misfires and what it means for us and our health. Our bodies face an invisible battlefield. Bacteria, viruses, fungi, and parasites, all of these organisms are all around us and even inside of us. Most are harmless, some are actually helpful, but there's a subset that we'll call pathogens that are true troublemakers. And that's not just microbes. We also have to defend against our own rebellious cells like cancer. Our defense comes in three different layers. First, barriers like your skin, tears, and stomach acid. These are your built-in wall and basically the moat that surrounds us. Then we have our second, the non-specific defenses, which are fast and blunt responses like inflammation and fever. And finally, there's the third line, our specific immune response, which is highly targeted and has memory. It's like going from castle walls to guards within the clubs to elite snipers who never forget a face. Pathogens come in many forms. Bacteria are living single-celled organisms, sometimes friendly, sometimes deadly. Fungi can be harmless molds or dangerous infections. Parasites are freeloaders with range from microscopic protozoans to worms. Then we have the viruses. Many are little tiny packets of genetic codes and prions, which these are bizarre misfolded proteins that can cause neurological disorders like mad cow disease. Each type possesses unique challenges for our immune system. Bacteria are prokaryotes. They don't have a nucleus. They are masters of survival, thriving in diverse environments, and many are essential to us, from helping digest food to making yogurt. But pathogenic bacteria can cause illnesses like tuberculosis or even syphilis. Luckily, antibiotics can be used to stop them, though resistance is becoming a growing concern. This sets the stage for why our immune system needs to be ready at all times. Viruses are much smaller than bacteria, and there is still a debate about whether they are truly living. They cannot reproduce on their own, and they hijack the host cell. Structurally, they contain either DNA or RNA surrounded by a protein called a capsid. Viruses cause many diseases, including COVID-19, influenza, hepatitis, rabies, Ebola, HIV, and even AIDS. Prions are misfolded proteins that trigger other proteins to misfold, creating a chain reaction. This damages brain tissue by filling it with protein debris and highly resistant to heat and chemicals. Prion diseases include Creutzfeldt-Jakob disease, mad cow disease, and chronic wasting disease in deer and elk. They are acquired through consumption of contaminated meats or, in rare cases, inherited mutations. The health risk of pathogens depends on three factors: transmissibility, or how easily it spreads from person to person, modes of transmission, which may be through respiratory droplets, fecal oral route, bodily fluids, or direct contact with virulence, which is the extent of the damage caused. When infections spread through populations, we classify them as epidemics. If they spread across countries or globally, they actually become pandemics like COVID-19. The lymphatic system has several key functions. It maintains blood volume by returning excess fluid to the cardiovascular system. It also transports fats and fat-soluble materials from the digestive system. Most importantly, for immunity, it filters foreign materials to defend against infections. The system includes lymph nodes, lymph vessels, and lymphoid organs. Lymph nodes are these small bean-shaped structures located along the lymphatic vessels. They act as filters, removing microorganisms, debris, and abnormal cells. Inside, they contain connective tissues, macrophages, and lymphocytes that actively fight infections. As lymph passes through the nodes, it is cleaned before returning back to circulation. Swollen lymph nodes often indicate an active immune response. The spleen is the largest lymphatic organ and is located in the upper left abdomen. It has two regions: the red pulp, which removes old and damaged red blood cells and stores blood temporarily, and then the white pulp, which contains lymphocytes that monitor for pathogens. Diseases like mononucleosis or leukemia can enlarge the spleen. Although it can be surgically removed, this does increase infection risk. The thymus gland sits behind the sternum just above the heart. It is where T lymphocytes mature and become functional. The thymus secretes the hormone thymosin and thymopoietin, which directs this process. It is largest and most active during childhood, then gradually shrinks and becomes less active with age. Tonsils are clusters of lymphatic tissues near the entrance of the throat that filter food and air. They can become inflamed and infected, which is tonsillitis. Adenoids are smaller tissues near the back of the nasal passages filtering air and that passes through. Both act as the first line of immune defense for inhaled or ingested pathogens. Our first line of defense includes physical and chemical barriers. The skin is the strongest barrier. Tears and saliva contain lysozymes, which is an antibacterial enzyme. Earwax and mucus trap microbes, while coughing and sneezing expel them. The stomach and vagina are acidic, which inhibit pathogens. Urination, defecation, and vomiting also helps flush out harmful organisms. Finally, resident bacteria compete with pathogens, preventing them from establishing. The human microbiota refers to all the microorganisms that live in or around our bodies. Many are beneficial and play roles in digestion, immunity, and overall health. Some can act as opportunistic pathogens if defenses are weakened. Probiotics are live microorganisms taken to restore or strengthen beneficial microbiota, while prebiotics are foods that encourage their growth. When pathogens bypass the first line of defense, non-specific defenses take over. These include the complement proteins, phagocytic cells, inflammation, natural killer cells, and interferons. These defenses respond to a wide range of threats without targeting specific pathogens. The complement system is made of over 30 proteins that circulate the body in an inactive form. When activated, they enhance immune defenses by marking microorganisms for destruction, attracting phagocytes through inflammation, and directly lysing invaders by puncturing their membranes. Inflammation is triggered by tissue injury and results in redness, warmth, swelling, and pain. Injured cells release histamine and other chemicals that increase blood flow and attract white blood cells. Phagocytes engulf microbes and clean up the debris, and tissue repair begins. Inflammation is essential for healing, but it can also be harmful if uncontrolled. Natural killer cells are lymphocytes that recognize virus-infected cells or tumor cells. They attach to the target, create holes in the cell membrane, and release enzymes that destroy the cell. This helps the body eliminate dangerous cells early in infections or cancer development. Fever occurs when macrophages detect pathogens and release pyrogens, which resets the brain's thermostat to a higher temperature. Normal body temperature ranges between 90 to 99 degrees Fahrenheit. We characterize a fever as anything that is over 100.4 degrees Fahrenheit. This makes the body less hospitable to microbes and strengthens the immune response. Normal fever can be helpful, but high fevers can actually be dangerous and cause tissue damage and must be monitored. The third line of defense is the immune response. Unlike nonspecific defenses, it recognizes and targets specific pathogens. It has memory, meaning it responds faster and more strongly after the first exposure. It can distinguish between self and non-self, and as well as between normal and abnormal cells like tumors. An antigen is a substance that triggers an immune response, often proteins or polysaccharides found in the surface of microbes. Our cells carry self-antigens called MHC proteins, which allow the immune system to recognize our own cells. Anything lacking these markers is seen as foreign and attacked. Lymphocytes, which are a type of white blood cell, are critical for specific immunity. B lymphocytes are responsible for antibody-mediated immunity, producing antibodies to neutralize antigens. T lymphocytes are responsible for cell-mediated immunity, directly attacking infected or abnormal cells and coordinating immune responses. B cells are activated when they recognize a specific antigen, often to help with helper T cells. Once activated, B cells divide to form two groups: memory cells, which remain for future immunity, and plasma cells, which secrete antibodies to fight off the current infection. Antibodies circulate the blood, lymph, and secretions. They bind to antigens and form complexes that neutralize pathogens, block them from entering cells, or mark them for destruction by phagocytes. Antibodies can also activate complement proteins, which help destroy that target cell. There are 5 major antibody classes. IgG is the most common and can cross the placenta to protect the fetus. IgM is the first antibody produced in an infection. IgA is found in the body secretions such as breast milk. IgD may act as a receptor on B cells, and IgE is important for the allergic reaction. Antibodies are Y-shaped proteins with 2 heavy and 2 light chains. The tips of the Ys form the antigen binding sites, which vary greatly in amino acid sequence, allowing huge diversity. This ensures that immune systems can recognize millions of different types of antigens. T cells originate from the bone marrow, but mature in the thymus. There are 2 main groups: CD4 cells, which become helper T cells, and memory T cells. Then you also have CD8 cells, which become cytotoxic T cells. Each type plays a distinct role in immunity. T cells cannot recognize antigens alone. They must be presented by antigen-presenting cells, such as macrophages or B cells. These cells display pieces of antigens on their surface alongside MHC molecules. When a T cell recognizes this combination, it becomes activated. Helper T cells secrete cytokines that activate other immune cells and are responsible for directing the responses. Cytotoxic T cells directly destroy infected or cancerous cells. Memory T cells remain in the body to respond quickly to future exposures of the same antigen. Helper T cells are activated when CD4 cells recognize an antigen on an antigen-presenting cell. Once activated, they divide and form clones. Some release cytokines that activate macrophages, B cells, natural killer cells, and cytotoxic T cells. Others become memory cells for future immune responses. Cytotoxic T cells, also called CD8 cells, target host cells infected with viruses or bacteria, as well as cancer. They also attack transplanted tissues if it is not matched properly. Once activated, they multiply into many cytotoxic T cells and memory cells. This action is essential for controlling viral infections and abnormal cell growth. Cytotoxic T cells kill by releasing 2 key proteins. Perforins create pores in the target cell's membrane, while granzymes enter the pores and break down the cell from within. Think of it like a grenade. The grenade goes in and everything blows up inside. Together, they trigger cell death. This ensures infected or abnormal cells are removed quickly. The immune system remembers antigens after the first exposure. The primary response takes several days, with antibodies level peaking at around 10 days. The secondary response is much faster and stronger, often presenting illnesses altogether. This memory forms the basis of immunity. Medicines support the immune system through immunizations, monoclonal antibodies, and antibiotics. Immunizations create long-term protection by preparing the body in advance. Monoclonal antibodies can trigger specific diseases directly. Antibiotics combat bacterial infections but are ineffective against viruses. Active immunization or vaccination exposes the body to a safe form of an antigen. Vaccines may contain killed or weakened pathogens, pieces of the organism, or genetic material. The body produces memory cells and antibodies in response. This primes the immune system for further exposure. Passive immunization provides pre-made antibodies for humans, animals, or lab preparations. It gives immediate short-term protection but does not create memory. It is useful for existing infections or when exposure is likely. Natural examples include maternal antibodies passed through the placenta or through breast milk. Antibiotics kill or slow bacterial growth by targeting unique bacterial structures such as the cell wall. Narrow spectrum antibiotics affect a small group of bacteria, while broad spectrum antibiotics affect many types but may disrupt normal microbiota. Antibiotics do not work against viruses. Antiviral drugs exist, but they are limited in number and actual effectiveness. Tissue rejection occurs when the immune system attacks transplanted cells as foreign. This reaction is mainly mediated by T cells recognizing mismatched MHC antigens. To reduce this, donors and recipients are matched for blood types and MHC compatibility. Immunosuppressive drugs are used but are increasingly vulnerable to infections. When the immune system responds inappropriately, problems arise. Allergies are exaggerated responses to harmless antigens such as pollen or food proteins. Autoimmune disorders occur when the immune system attacks the body's own tissues. Both situations demonstrate how precise immune regulation must be. Allergies occur when the immune system reacts to allergens such as pollen, mold, dust mites, or certain foods. These allergens are harmless, but the body responds as if they were pathogens. The reaction can cause mild symptoms like sneezing or severe reactions that affect breathing and circulation. Anaphylactic shock is the most dangerous form and can be life-threatening. Many times people get allergic reactions and have hives, which of course again can be mild or can also be very life-threatening. During the first exposure, allergens activate B cells, leading to the production of plasma cells that secrete IgE antibodies. IgE binds to mast cells and basophils, priming them for later reactions. Upon re-exposure, allergens trigger these cells to release histamine and other chemicals. This leads to allergy symptoms such as inflammation, swelling, and itching. Allergic reactions can be localized or systemic. Localized reactions affect only the exposed areas, such as the nasal passages in hay fever. Systemic reactions affect multiple organ systems and can cause widespread inflammation. Severe systemic responses may result in anaphylaxis, which requires immediate treatments. Allergy treatments include antihistamines, which reduce mild symptoms by blocking histamine. Severe cases of anaphylaxis are treated with epinephrine or the EpiPen, which quickly restores breathing and blood pressure. Allergy shots may help desensitize the immune system over time. These treatments aim to reduce symptoms and prevent life-threatening reactions.